In our last video, we looked at many of the impacts of natural selection. We looked at overdominance and underdominance. We looked at directional selection. We talked about the influence of genetics and how genetics can play a role in rates of allele change and rates of fixation. All that is frequency independent natural selection, or we usually just refer to as natural selection because that was the first type that was really investigated and that Darwin was talking about. However, there are also some very interesting examples of what we call frequency-dependent natural selection. And in frequency-dependent natural selection, your fitness depends not only on your own characteristics, but on how common those characteristics are across the population. So there are two directions for frequency-dependent selection. The first is called positive. And in positive frequency-dependent selection, you do better if there are more individuals that are like you in the population. And so your fitness is dependent not only on your phenotype, but whether that's a common phenotype or not. The best example of that is aposematic, sometimes pronounced aposematic coloration. And aposematic coloration is when an organism displays bright colors as a warning to let predators know that it's toxic or in some other ways dangerous. Okay, and so red poison dart frogs with bright colors are an example. The monarch butterfly has distinct wing colors that are a warning to its bird predators to stay away because it's noxious, it's toxic, and the birds do not do well when they eat it. Now, think about this when it first evolved. Now, most frogs are green or brown and kind of blend into their background, and that's a good adaptation to stay away from predators. But if you're toxic enough and then have a mutation that makes you stand out, that might actually be a good thing because predators can learn to stay away from you. But if you are one of very, very few frogs that is bright red and everyone else is blending in, that's probably not a great thing because even if you're toxic, it doesn't do you any good if, a naive bird or another predator that doesn't know any better eats you, you're still dead. And so there's still a little bit and some work that in individual examples that need to be done to figure this out. But how do we get over this initial bump where we need a certain level of representation in the population? But once you reach that level, then suddenly it's a really, really good thing, and it's better to be standing out because birds learn fairly quickly to stay away from the toxic ones. They don't get as much predation as the green ones and the brown ones in the population, and soon that entire population is going to have bright colors. The same thing occurs with monarch butterflies and their orange wings. And there are other examples, salamanders that are toxic show this. So aposematic coloration is a good example, but you do well as long as everyone reaches many people in the population have the same trait. And so the eventual outcome of positive frequency dependent selection is fixation of that beneficial allele. As long as we can get over that initial hump of getting enough individuals in the population so that the positive method or the positive part of that takes over. Now, the other example is negative frequency dependent selection. Now, I'm going to use poker as an analogy here because I think it works really, really well. Is bluffing in poker, if you're familiar with the game, is bluffing a good strategy? Now, the short answer is yes, as long as you don't do it too much. If you bluff all the time, then pretty soon it's going to be obvious what you're doing and your bluffs are going to get called and you're going to lose all your money. And so negative frequency dependent selection is kind of the same thing. It is a strategy that works well as long as it's not too frequent in the population. Now, there are a couple examples here that your book uses. I'm going to use this one on the left, which is an example of a very interesting physical trait that we'll use later in the class also. And this is something that we call sneaker males. So just a little bit of background. And this occurs in other organisms, occurs in some fish also, but the beetle's kind of a fun example. This picture here on the left is a picture of two male dung beetles. Now, dung beetles, if they're going to reproduce, as their name implies, they need to gather a ball of dung and then lay eggs on it for the babies. And in many species, the male gathers the ball of dung and then females will come in and he will mate with those females. So that's a way that he can maximize his fitness. And the big males that have these large horns are very, very competitive, and they will find the dung, they will guard it, sometimes they'll even bury it, and then they'll keep all other males away, and females will come in, and that's a very successful strategy. In some species, they have evolved these males that look a little bit like females, are smaller, and have very different behavior. So they don't seek out dung, they seek out other males who have already done all of the work. And then they will dig a little back entrance to the dung chamber, right? The male's guarding the big front entrance, only letting females come in, mating with them, and he's being very successful doing that. But these sneaker males will sneak in the back way, and then as the females come down to lay their eggs, he will mate with those females, and some of the resultant offspring will be his. Now, is being a sneaker male a successful strategy? And the answer is yes, up to a certain point. Because at the beginning, when this trait first evolved, those first males did really, really well. There were lots of big wild type or the initial type males guarding the dung. They had lots of places they could go and work their strategy, and it was very successful. But as more and more offspring began to carry this trait, more males going around looking for the big guarding males, then it suddenly becomes less of an advantage. Because you have to compete with more individuals like you, there are fewer of the big males to take advantage of. And so this is negative frequency dependent selection because it works well as long as there are low frequencies. The inevitable result of negative frequency dependent selection is that you will reach a balance point. And at a certain point, it no longer becomes good or as good to be a sneaker male. And so we reach a balance in the population. And so you'd have something that looked like this, where you would have some number of the bigger phenotypes, some number of the smaller phenotypes, and they would reach some sort of a balance point. Now, there are many, many more complex things that can impact traits, and particularly when we are looking at two traits together. Up until now, we've been treating traits as if they are completely independent. And that's actually a pretty safe assumption for the vast majority of traits. So for example, if I was to suddenly become supreme dictator of the world, and that's a really bad idea to make anyone, no matter how benevolent they are, supreme dictator of the world. So let's say you did it, and suddenly I got it into my head that I would really like it if we had unattached earlobes, if everyone had unattached earlobes, right? Earlobes that dangle down rather than go to the side of the head. It's a common trait that's used in genetics class. It's a fairly simple genetically controlled trait. So if I decided that and I only allowed people with dangling earlobes to reproduce, I could change the frequency of that trait in the population. And if it was fairly simple genetics, I could maybe even completely get rid of attached earlobes and have the entire human race with dangling earlobes. Okay? Obviously, that's kind of a dystopian future bad scenario. All you people with attached earlobes are saying, wait, what's going on? But we could do it. In theory, it works. It's value-wise, right? The science does not help us decide whether things are moral or not. That's hopefully, by most people's standards, a very amoral thing to do. But if it was done, think about what impact I would have on the average height of the human population. So are dangling earlobes and height correlated? And the answer for the vast majority of traits are no. There's no correlation. They get completely mixed up because of random assortment during meiosis. However, there are instances where traits can be correlated together. Now, I've listed two here. This is actually a mistake on the slide. I forgot to change it before I uploaded it. So actually, there's only one cause that we're going to talk about. There are others too, but this is the only one. Traits can be correlated without genetic linkage if they need to be together to provide an advantage. So they're an adaptation as long as they're together. But if they're separated, then they're not. So let's look at this where we have two traits. So this is an example in snakes. Now snakes, when they are faced with some sort of a threat, have two options. They can hide or they can flee. Now if you hide, there are certain patterns on your skin that are going to help you blend in better. If you flee, there are other patterns that actually help kind of break up your profile as you're slithering along. That makes it harder for your prey, a hawk or maybe a mammal coming along trying to eat you. Makes it harder for your prey to visually pick you out and attack you while you're moving. So here's an example where a behavior, if it is with the right color pattern, is a benefit. But if those two traits are separated, so if you have the fleeing behavior but the hiding color pattern, you're not going to do very well. And if you have the hiding behavior but the fleeing color pattern, the reverse, you're also not going to do very well. So in these cases, we're going to have natural selection that tends to put these together. And particularly if there's other forces in the environment that separate these two, we might have now another thing that creates a speciation force because we're separating the population into two distinct groups. But genetic linkage also causes traits to be correlated. And genetic linkage is very simple. It's simply when two genes are found really close to one another on a chromosome. And so what that means is the only way to break up the genetic linkage is if there's a crossing over event. So random assortment completely breaks up and randomizes alleles that are found on, genes that are found on different chromosomes. But if they're found right next to each other, it really can't break them up unless there's a crossing over event. So random assortment has no impact on genes that are on the same chromosome. But we have a second force of recombination, crossing over. The closer two genes are to one another on the chromosome, the less frequently they'll be broken up by crossing over. So we call that genetic linkage. Okay, we're going to come back to genetic linkage in a little bit. But I just want to give you definitions and maybe just a little tiny bit of information about some other genetic things that can play a role. Epistasis. So epistasis is the unpredictable interaction or the complex interaction of alleles at different sites. Don't get it confused with epigenetics. That's something very different. But epistasis is this unpredictable interaction of alleles at different sites. This coloration pattern and the MC1R gene, which is a pigmentation gene in mice, is one example of that. Don't look at all of the details. Just know the definition for epistasis. And that can complicate natural selection and make it somewhat linked with other traits. Another thing, of course, that makes it very complex are these continuous variation traits, right? And continuous variation, like height in human, or in this example, coloration, the amount of pigment in the seeds of wheat, excuse me, those things can make natural selection more complex. Now, we call this continuous variation or continuous trait. And it's almost always the result of polygenic traits, meaning that there are many, many genes. In this case, we've got 3 different genes. Each of those genes has 2 alleles, and all of those contribute to the coloration of the wheat. Something similar goes on in pigmentation in humans. There are multiple genes, not just 2, but multiple genes that impact a certain trait, and each of those genes might have many different alleles. Now, this fits very, very well into Darwin's initial hypothesis and ideas about evolution, because the evolution is going to, natural selection is going to be able to work on this situation, but it's probably only going to have very little changes each generation. And so these complex traits mean we can shift things, but it might take many dozens of generations, which is what Darwin was talking about, those very, very slow changes that we see in evolution. And then also, just by excuse me, guys, just by looking at a population, another thing that can impact natural selection is latent variation. So just by looking at a population and measuring it, we often miss some of the genetic variation that's there in the population. Now, latent variation is not as much of an issue these days because we have tools and the ability to look at all of the features by DNA sequencing and taking large samples. And so latent variation is not as much of an issue, but it can still be present. So for instance, we might miss recessive alleles if we only look, if it's a rare recessive allele especially, if we only look at the phenotypes. If there's epistasis going on, we might miss some of the different combinations that are possible because of these unpredictable interactions of alleles. So just be aware that often there's a lot more variation than is easily measurable in a population, and we call that latent variation. Now, let's go back to that issue that we talked about, genetic linkage. So I'm going to introduce one more word here that you need to know, a haplotype. A haplotype is basically like an allele, but it extends the definition of allele to things that don't have an impact on phenotype. So a strict definition of allele is a variant of a gene that has some sort of phenotypic difference. A haplotype is any variant at all. So if I had a silent mutation that made no difference physically, so I mutated someone so they were still O blood type, but they had a C at a third position in that gene instead of an A, it was still the same amino acid, that would be a different haplotype. We can measure it if we sequence the DNA, but we wouldn't see any physical manifestation of it. So just be aware, and we'll use the term haplotype sometimes. It's a little bit of a broader term, but in many cases is very similar to the word allele. And so if we have physical linkage of haplotypes or different alleles, then they're almost always going to be inherited together. So here notice that if I have the A gene and the B gene on different chromosomes, I get this nice predictable interaction. And this is what Mendel was doing with his Ps. And I get 25% homozygous dominance. I get 25% homozygous recessive. And then I mix up these others. But if we have those on the same allele, or sorry, on the same chromosome, so the dominant alleles are on one chromosome and then the recessive alleles on the other, they're always going to be inherited together unless we have crossing over. And so with crossing over, we get this imbalance in these nice predictable frequencies that Mendel did. And so we call this imbalance linkage disequilibrium. Now, we're not going to measure this or predict it quantitatively, but you just need to understand it. So the reason that we get linkage disequilibrium, the reason that alleles are linked, are a couple things. If it's a very recent mutation, then it's going to be linked right next to whatever allele it just happened to pop up with. So if we get a mutation in the A gene, and it's just right next to a big B allele, then that little a, that new mutant, is linked to the dominant allele, the big B. Natural selection can also cause it if we're pushing for two traits to be selected very, very rapidly. Migration from another population that is not diverse can cause genetic linkage also. And then finally, genetic drift, especially in extreme cases if there's very, very small populations. So all of those forces of evolution can, if under the right conditions, cause genetic linkage. And the only way to dissipate it or break up genetic linkage is via crossing over. And so here's an example of that. So here is what we would normally predict for, this is a more complex Punnett square where we're looking at 16 different possible outcomes because we have two genes. And these are all the possible outcomes, and we would predict they would be completely in the same amount if they were found on different chromosomes. But because they are found on the same chromosome, we're going to predict instead we're never going to be able to get these recombinants. We can never get a big A, little b, a little a, big B, because notice, big A, big B are on the same one. Anyway, the linkage here works out so that we can never get these two genotypes. However, when we run this experiment, often we'll find very tiny, small numbers of those rare recombinant genotypes, so represented here by the square size. We get this rare recombination occurring. And so with no crossing over, that's impossible. But with crossing over, every once in a while, we're going to mix that. If that happens over a long enough period of time, eventually those alleles get completely mixed together, so there's no longer an association, there's no longer a linkage of one allele with the another allele. So over time, crossing over will break up linkage disequilibrium.